Well, thank you for your patience. Good morning. I'm Jim Roberts. I'm a technology licensing officer at the TLO here at MIT, and I work very closely with our first golden speaker this morning, Ariel Furst. Ariel Furst received her Bachelor of Science degree in chemistry from the University of Chicago, working on the chemical synthesis of proteins. She completed her PhD at the California Institute of Technology, developing new cancer diagnostics based on DNA charge transport. She was then a postdoctoral fellow at the University of California, Berkeley, and now Ariel is the Paul M. Cook Career Development Assistant Professor of Chemical Engineering at MIT. Ariel is passionate about STEM outreach and increasing participation of underrepresented groups in engineering. She invents technologies that improve human and environmental health by increasing equitable access to resources. Her lab develops technologies that solve problems related to healthcare and sustainability by harnessing the capabilities of biological molecules and cells, and based on their understanding of the biological redox process. She is a founder of two startups, SeaBio and Helix Carbon, and you'll hear from her about those today. As a TLO, I've had the privilege of filing several patent applications with immediate commercial potential for Ariel, and they include CO2 capture and conversion, DNA electrochemistry, cost-effective point-of-use biosensing, metaphenolic network coatings, microbial degradation of pesticides, electroactive microbes for sustainable technologies. You can see her interests are very wide-ranging. So Ariel will speak for the better part of the remaining hour, and we'll answer any questions afterwards. We invite you to stay for a short reception afterwards with her, and if she's able to stay, and with other members of the TLO as well. And I also want to let you know that we have another golden speaker next Tuesday at the same time, 10 in the morning here, and that will be Sertak Karaman, who is with the Astronautics and Aeronautics Department. So I'd like you to welcome, or help me welcome, our first golden speaker, Ariel Furst.
Thank you. Thank you, everybody. So today I figured I'd tell you a little bit about my path to being an entrepreneur. It wasn't something I thought I was going to do when I was getting my PhD, and it's really the ecosystem at MIT that's enabled me to commercialize technologies for my group. So just to make sure everybody's awake this morning, what do we notice about this picture of Earth at night?
It's lit up.
It's lit up, but only in certain places, right? So there are some regions where there's a lot of electricity and other regions where there's little to no electricity indicated by the amount of light. Generally, the global south is pretty dark compared to developed nations. And this is because about 1 in 10 people globally don't have access to electricity still. This means that about 2.5 billion people rely on dirty fuels to heat their homes and cook their food. And so this is really what motivates my group, is the idea of energy equity for sustainability, environmental remediation, and human health. So has anybody heard of the concept of energy equity before? I see a couple nods. This is good, because there's a whole row of my group here, so they should all be nodding. This is basically just the concept that disadvantaged groups have been disproportionately impacted by the negative impacts of things like industrialization. And so we see it as our job as researchers to develop technologies that support these groups. When we think about kind of inequitable technologies, one of the examples I like to use is a Tesla. It might be great for reducing emissions, but if only 0.1% of 0.1% of 0.1% of the global population can access this technology, how big of a difference can it really make? And so my group is motivated to make accessible and inexpensive technologies that can be broadly used. So I wanted to talk a little bit about my background first. As I mentioned, entrepreneurship was not something that was on my radar before coming to MIT. So how do we think about problems, and how did I kind of get to where I am? Well, we're going to have another quiz now. So basically, since humans have been building things, we've been taking inspiration from natural systems. This is a suit of armor from 1500s. AD, inspired by the scales of the pangolin. Quiz time. Does anybody know what this is? It's swimsuit material from the 2010 Olympics that has since been banned because it gives swimmers too much of an advantage. And it's based on the structure of shark skin. How about this one? Yes, nice to, yes, it's Velcro. And Velcro, which we think of as something that's totally human-derived, right, is actually based on the interaction between seed coatings and animal fur. So when we think about the built environment, we can really trace a lot of the innovation there back to natural systems. And so that's what my lab focuses on now, but that's not actually where I started. So as anyone who does research in here knows, most of the most exciting things in research come out of multiple failures. What got me interested in research was not chemistry class in high school. I actually hated science class. It wasn't until I had the opportunity to work in a lab where the project was pitched to me as trying to figure out ways we could use proteins to power our cell phones with vodka as an input. What high schooler doesn't want to figure out how to power a cell phone with vodka, right? Unfortunately, this didn't work, but it was the start of what I saw as the excitement in research, which is not knowing the answer to the question. So then I moved on after undergraduate to my PhD and postdoc in California, where in my PhD, we thought about how we could use DNA as more than just genetic material. We could use its physical properties to give it additional function. It can act like Velcro, and it can also act like a wire. And then in my postdoc with Matt Francis at Berkeley, he thought about how we could use protein materials, specifically the coatings around virus particles, as things like drug delivery vehicles. So in both of my experiences, these were fundamental scientists not thinking about engineering necessarily, but really taking what biology has given us and applying it in different ways than we normally think about. After that, came to MIT, started my group. This is our most recent group picture. And what we like to focus on is how we can think about things that are easy to implement. And that's really been one of the most rewarding things about MIT is learning how we can take what we do in the lab and translate it to something that's actually commercializable. So the motivation for our first company, SeiaBio, was nothing to do with what the company looks like now. So I figured I'd just talk a little bit about the pathway to how Seia got to where it is. So what we were originally thinking about in my group is how we can study microbial biodiversity. So along with climate change, loss of biodiversity is one of the major negative impacts on our globe right now. And as we can see, we've lost a significant percentage of the global number of species. These are basically the number of species that have gone extinct since 1970. And importantly, microbes really are the basis for all life on Earth. They cycle nutrients in soil. They help protect both plants and animals and humans from external pathogens. And they help us metabolize our food in our guts. And so as we lose that biodiversity, we become more susceptible to disease. Our soil becomes less healthy, and we are less able to sustain our global population. So we wanted to take microbes from the environment and study them in our lab. The problem is soil is a nice, cozy environment for the microbes. When we take a sample of microbes from the soil and try to transport them into the harsh environment of a lab, we lose about 90% of the species. And so our goal was to figure out a way to just take microbes from their natural habitats and bring them to our lab. What we decided to try was a self-assembling material that forms on the outside of individual microbes and kind of acts like a suit of armor. You can also think of it like the coating on the outside of the M&M that helps prevent the insides from melting. So what are these suits of armor made of? Well, We wanted to think about things that were inexpensive and easy to access. And if we were going to be using them on microbial samples that were coming from things like soil, we also wanted them to be food grade. So we found materials that were derived from polyphenols, which are things like green tea extract or tannins from coffee and tea, as well as vitamins and minerals that you take every day, things like iron and manganese that are found in supplements. And when you mix these two things together, they self-assemble into this very thin coating that forms on the outside of microbes. And these are called metal phenolic networks because they're made of polyphenols and metals. And I'll be referring to them as MPNs. And these are really cool because they form really easily just by mixing these components. So if you watch this video here, this is just the microbes being coated. All we have to do is mix the metal with polyphenols and microbes, and that blue color is the coating forming. And as we change the components of these coatings, we change the physical properties of those coatings. And that's what gives us these different colors shown here. And if we image microbes after they've been coated, we can see that these coatings form homogeneously on the surface. And we think that this is really important for actually protecting the microbes. So this is just an uncoated microbe. And this is a microbe that's been coated with a layer of those metal phenolic networks. And by TEM imaging, we can see that the metal ions here are what's giving us the kind of sharp contrast. And that microbe is fully coated. And because these coatings are very thin, they're about 2 to 10 nanometers thick, we can actually coat these subcellular structures. So this really allows us to protect the whole microbe. And what we see is that when we coat these in batch, so obviously we're not going to do this one microbe at a time, we get uniform coatings across all of the microbes. So this is just a stain that we've put on the coating, and you can see here the shell structure that forms. And each of the microbes in this matrix is completely coated, and they're all evenly coated. And what we found is that these coatings protect against pretty much every stressor we could think of throwing at them. This ranges from things like oxidative stress, putting them in hydrogen peroxide, UV and radiation exposure, temperature fluctuations, chemical exposure, mechanical stressors, and high salinity. And so we thought, well, maybe this could be useful for things beyond just preserving microbes to bring them back to the lab, right? Maybe we could actually use this to improve human health. How many people in here take probiotics regularly? Awesome. About 99.9% of the microbes in that are dead. And that's because there are a few species that we know how to protect really well, but the manufacturing process is very harsh on them. And the problem is probiotics companies are basically marketing companies. And so they don't care how many live microbes they give you. They just want you to buy their product. And so we know how to manufacture these. We get about 0.001% viable cells. But there are many microbes that we think could be beneficial for treating things like Crohn's disease, IBS, and other kind of diseases that would require an FDA-approved treatment, but we can't actually manufacture these because they're very delicate. So what we thought initially for our metal phenolic networks was that we could actually use them to help us move beyond the probiotics you buy at the grocery store. And so this is when we got involved in the MIT ecosystem. We originally submitted a grant on this to the Deshpande Center. This gave us some seed funding that allowed us to take these metal phenolic networks coatings basically from fundamental research to where we could have an MVP, where we could have a minimum viable product. And this is where I met Leon Sandler, who used to be the director, and I also met Jim Roberts in person. And that is actually one of the best things about the Deshpande grants is you actually get to sit once a quarter with your technology licensing officer and discuss the progress on the project and IP strategy. We then decided to do the I-Core program. This is an NSF-funded program that basically teaches you how to do customer discovery. So Roman in the Martin Trust Center runs a New England version of this, so it's a kind of mini version. So we did that, and then we did the full version, which is a very intensive 7-week course during summer, where we had Ann Madden, who's a badass microbiologist who's worked at a variety of startups, including Indigo Ag, as our industry mentor. So this program is a full-time program where you have to complete 150 customer interviews in 7 weeks. You have an industry mentor who helps make connections, and it's really kind of the best way that we've found to train ourselves as scientists to be entrepreneurs. I then got very lucky because the first cohort of the Future Founders program was starting right as I-Corps was ending. And so Kit Hickey from the Martin Trust Center and Sangeeta ran this. And along with these amazing women, I got another industry mentor and got to spend a semester digging in on what we thought was our beachhead market. So our initial beachhead market was going to be living biotherapeutics. This is a fancy word for FDA-approved microbes. So these are different from probiotics because they would actually have to go through clinical testing. And part of the reason that this is only a $1 billion market right now is because microbes are really hard to manufacture. So we can't actually make them to do clinical testing. The original company name for this was Farmore. And this is part of the reason why it's really important to talk to marketing people and think about your branding. We later changed to SEO, which I'll talk about. But basically, what we found through customer discovery was that about 60 microbes had gone to preclinical trials. Only 25 had made it to phase 1. And in phase 1, you don't have to have a final formulation. So here they were basically making patients drink microbes in the stuff that the microbes were grown in. It's pretty gross. Phase 2 is when you need a final formulation. So you see that pretty big drop off. Only 5 have moved to phase 3, and currently there are 0 FDA-approved living biotherapeutics. And again, this is because these microbes have to be alive in order to be efficacious. And there's so much batch-to-batch variability that they can't even get consistent batching for clinical trials. So our initial targets for this were 2 microbes, B theta iodo-omicron and B subtilis. B theta is an anaerobe, and it's found in healthy people's guts in very high amounts, and it's significantly depleted in people with IBS or Crohn's disease. And so it's been hypothesized that if we deliver this to your gut, it could help treat those diseases because it's known to secrete anti-inflammatory compounds. The problem is this microbe is what's called a facultative anaerobe, which means if it's exposed to oxygen, it dies. Most of our manufacturing plants are full of oxygen, right? In fact, in the US, there's only 1 plant that has end-to-end manufacturing that's totally anaerobic, and it costs about 10,000 times as much as standard manufacturing per batch. So it's prohibitively expensive. Additionally, B subtilis is often found as a probiotic, and this is easy to manufacture because it's sporulates. So you can formulate it as a spore. It's very hardy. The problem is, it is so hardy that it goes through your entire digestive system as a spore. So it has no benefit. So we wanted to see if we could actually protect this in its vegetative state so it might implant and actually be useful. B subtilis is also important because not only is it prevalent in healthy microbiomes in our guts, but it's also prevalent in healthy soil microbiomes. And this is something we consistently saw as we learned more and more about the composition of microbiomes. What's healthy for our guts, a lot of those species are the same ones that are beneficial in the soil. So as we learned more and more about living biotherapeutics, we realized that this was really going to be a pretty niche market. And it could help people with things like IBS and Crohn's disease, but there were bigger issues we wanted to tackle, especially when we think about global equity. So by 2050, it's anticipated we're going to need about 50% more food to feed the global population. And currently, most farming, especially in the United States, is done with chemical fertilizers. These have been fantastic for feeding the global population. Right now, it's estimated that the addition of chemical fertilizers feeds about half of the global population. So it's enabled Earth to sustain a global population above 8 billion people. The problem is its production causes a significant amount of greenhouse gases. About 2% of global greenhouse gases are just because of the Haber-Bosch process for nitrogen fertilizer production. It also damages native ecosystems, and a lot of it is wasted as runoff. And not only that, but where we're concerned, for farmers, it's increasingly expensive. And so farmers are now having to decide between applying the optimal amount of fertilizer or buying the right number of seeds or watering efficiently because their margins are so slim. Well, in nature, microbes do everything that chemical fertilizers do. So they can balance soil nutrients, they can improve crop stress tolerance, they can even secrete pesticides. So in theory, if we can formulate the right microbes, we can pretty much completely negate the need for agrochemicals in general. That's everything from fertilizers to pesticides. Not only that, but when people have added these microbial products to their farmland, even when they're still using chemical fertilizers, they see improved crop growth and yield. The problem, as you've probably gathered at this point, is that the microbes have to be alive. And just like our guts, much of the soil is not exposed to oxygen, and so these microbes don't like oxygen, they're very delicate, and when they're not in their native niche, in their native home, they're very hard to grow and manufacture. So when we thought about moving beyond human health, one of the microbes we wanted to target was Pseudomonas chlorophyllus. This is a really heat-sensitive microbe, so if you heat it above room temperature, about 25 degrees Celsius, it dies. That's a problem, right, for any sort of distribution, because you either then need cold chain, which is expensive and energy intensive, or you significantly reduce your yields. But this microbe is really cool because it can pull nitrogen gas out of the atmosphere and turn it into ammonia. So it can produce chemical fertilizers on site without any excess energy, without high temperatures and pressures, and it can also secrete antifungal compounds. So it can act as both a natural pesticide and a natural nitrogen fertilizer. And so this is when we pivoted and moved our focus of our company from what it was at Farmer, which was human health, to sustainable agriculture. Saya is the goddess of bountiful harvest, so we moved towards a more kind of holistic approach to the company, and this is our initial team. Timothy Moy, who is a 2024 EMBA here, is the CEO and co-founder, and we recently hired a third co-founder, Heidi Arches, who's also the head of our R&D. And the focus of Saya Bio is to broadly enable the use of microbes. So we've talked a little bit about human health. We can do things like living biotherapeutics. We can also make vaccines more accessible. So if we don't need cold chain, more people can access vaccines. We can also do things like replace fertilizers or do enhanced weathering, which is basically a fancy word for doing direct air capture of CO2 and sequestering it in soil. We can develop new building materials using microbes. And we can move beyond chemical synthesis. So if we replace chemicals with microbes for synthetic processes, we can decrease the amount of solvents we need, decrease the amount of waste we produce, and overall kind of improve the sustainability of our chemical ecosystem. And so as I mentioned, we can protect against a variety of stressors. So B theta is that gut microbe that doesn't like oxygen, right? So what we were able to do is to take this guy, coat it in our metaphenolic networks, and leave it on a benchtop. When we then tried to grow it, you can see that our coated cells grew, while, as you would expect, all of our uncoated ones died. So we've now enabled the production of a microbe that couldn't be produced before. Similarly, BCG is the tuberculosis vaccine. It's a microbe. It's basically the cow version of tuberculosis. The problem is it needs cold chain storage right now. And the majority of where this vaccine is distributed is in places like India, where it's very high heat and humidity. And as you can see here, we can store this guy at 50 degrees Celsius, that's about 130 degrees Fahrenheit, at high humidity for a couple weeks. And so what this allows is places that don't have constant access to things like electricity or cold storage to now maintain a supply of this vaccine. As I mentioned, we're also interested in ag, so P. chlororhynchus, that's that guy that can both basically fix nitrogen in the soil and make pesticides on site. We also stuck this guy in an oven at 50 degrees Celsius and 48% humidity. These are conditions that are prevalent in storage in places like India and Brazil, where they can't currently store chemical fertilizers because of the explosion risk that that high heat makes. So as you can see, when we stick these guys in an oven, we have multiple different formulations that all protect, with one that protects even better. And as you would expect, the uncoated ones are all dead. What was really surprising to us, though, is the impact that these stressed microbes had on germination. So if we look at germination of all of these different crops, which is what the students who were doing this study wanted to take home at the end of the study, we can add either just water, this is our control, fresh microbes, so this is the standard for regenerative agriculture right now. These have to be grown on site, so it's limited to very wealthy farms. We can also take freeze-dried microbes that have been stressed, and so we would expect most of these to be dead, or are coated in stressed microbes. And as you can see, across the board, we have about 150% higher germination at 10 days with our coated stressed microbes, even than with fresh microbes. So this truly is an enabling technology for the broad use of microbes. As I mentioned, we can also use this for the built environment. So this is work by Yuk Young, my postdoc, who's looking very embarrassed right now. Microbes can basically take CO2 from the atmosphere, combine it with a magnesium or calcium ion, and make limestone. And so this allows you to do direct air capture while also making tough materials. The problem with these right now is that if we try to incorporate microbes into things like cement, in order to protect those microbes, we have to use squishy materials usually. Think about like a gummy vitamin. That's effectively what people are using. So that destabilizes the cement, right? So now we can only use this for decorative purposes. But we know that with our coatings, we can dry microbes, we can pretty much do whatever we want to them, and they'll survive. So now we can put these microbes into a cement mix. This is a 10 centimeter by 10 centimeter slab of cement. And after 26 days, we get 40% crack healing. So we've been able to actually heal macro scale cracks by just incorporating 0.1 weight% of these microbes. And so basically, what SEA has now is a portfolio based on this platform technology that allows us to use microbes across all these different industries for everything ranging from human health applications to agriculture to CO2 capture and utilization. And so that was when I realized that I very much enjoyed entrepreneurship in general and got exposed to some additional resources on campus. So 15360 is Bill Ouellette's class, Disciplined Entrepreneurship. And 15366 is Climate and Energy Ventures, which is a team-based class where groups of MBA students basically work on projects pitched by faculty. So it's a fantastic resource if you have a technology but are not sure how it would fit in a commercialization strategy. And this is where Helix Carbon was derived from. Teams from 15360 and 15366 ended up merging and co-founding this company. And the fundamental basis of Helix Carbon is thinking about CO2 from a chemical engineering perspective. So if you shove enough electrons into it, maybe it could be something profitable and useful. And we can see that here. So CO2 per ton market price is obviously not very profitable, but if we can basically reduce it enough to generate things like carbon monoxide, which is a component of syngas, formic acid, or ethylene, that price per ton makes it a valuable asset. And the way we want to do this is using electrochemistry. So electrochemistry basically allows you to take clean energy inputs, things like solar or wind, mix them with CO2 and water, and make things like ethylene, which are starting materials for polymers and aviation fuels, which are going to be tough to decarbonize industries. So why isn't everyone who's an electrochemist on campus doing this right now? Well, the problem is because we do this in water, the amount of energy we're putting in to turn CO2 into things like carbon monoxide is pretty similar to the amount of energy it takes to split water and generate hydrogen. So a lot of our energy in these processes is not going where we want it to. It's generating hydrogen rather than reducing CO2. So going back all the way to the beginning, if we remember thinking about bio-inspired materials, we thought, well, maybe if we look to natural systems where we have things like enzymes that function very specifically and selectively with very high efficiency in water, we can improve the ratio of CO2 reduction to water splitting. And so we looked to small molecules, basically. And based on my previous work using DNA, we thought, well, maybe we could harness these unique physical properties of this molecule to improve that ratio after catalysis. So if we think of DNA not just as genetic code, but as these two individual strands that can come together, it's basically like a Velcro, right? If we attach one thing to one sequence and something else to the complement, they will naturally come together without any additional energy. And so what we found is that if we just scaffold known catalysts with DNA, we decrease our overpotentials by more than 30%. So our overall system energy consumption is about 30% lower. This translates to about a 25% lower system cost. This is also because we no longer need precious metal catalysts. We get about 90% product purity, so we're generating about 90% carbon monoxide and only 10% hydrogen, which for carbon monoxide is fine because the other component of syngas is hydrogen. And when we take all this together, at the current electricity prices, we can make syngas for $350 a ton. Current pricing for fossil fuel-derived syngas is about $700 a ton. So what this means is that people who would use this technology to capture CO2 and convert it to syngas would not only be doing it for sustainability, but also for profitability, which really seems to be the only way to move forward with sustainable technologies. And so what we see, this is just the data from that, is higher stability. So we don't need to change out our catalyst as frequency. We decrease our overpotential while also increasing basically the number of molecules we get out for the energy we put in. And we speed up this reaction by about 1,000-fold. So we're generating significantly more product that's more pure with way less energy input. And so I just wanted to introduce the Helix Carbon team. Evan is a 2024 MIT MBA. David is a 2025 MIT MBA. We have an amazing mentor from Deshpande funding from this. And these have been our funding sources so far for Helix Carbon. And that is what I had to tell you about today. This is the story of the first two technologies from my lab that have spun out. I hope you've seen the connection in how we approach challenges in sustainability, which is to think about how we can generate technologies that are affordable and accessible. And I also obviously need to thank my lab. I started in 2019, so many of my first graduate students joined the lab without having met me in person. We've had an amazing team, many of whom are here and who you can talk to about their experience during the networking session. We've also had an amazing group of undergrads. Currently, our lab has graduated 60 year ops. So we've had an amazing experience with them, and they've significantly contributed to a lot of the work I've talked about today. We also have had amazing high school students and amazing interactions with people on campus, like the TLO office, as well as groups like JWAFs that were some of the first to give us funding. So thank you, and I am happy to take questions.